What is the role of RNA polymerase II in eukaryotic gene expression, and how does it interact with other transcription factors to initiate transcription? Answer: Na polymerase II purament is a primary enzyme responsible for transcribing mRNA in eukaryotic cells. Its activity is essential for gene expression as it synthesizes messenger mRNA from DNA templates. The process begins with the binding of general transcription factors GTFS to the core promoter region, usually containing a TATA box. The transcription factor TFIID binds to the TATA box, which leads to the recruitment of other GTFS and RNA polymerase II. Transcription is regulated by the binding of enhancer and silencer sequences. Enhancers, which can be located far away from the gene, interact with transcription factors to increase transcription levels. Silencers, conversely, reduce gene expression. These regulatory elements are bound by activators or repressors, which interact with the transcription machinery to either promote or inhibit transcription. Post-transcriptional modifications, including mRNA capping, splicing, and polyadenylation, also help in stabilizing and processing the transcript for translation. Thus, our RNA polymerase II works in a highly coordinated manner with other factors to ensure efficient and accurate gene expression, which is crucial for cellular function and organism development. In a clinical scenario, a patient undergoes genetic testing and a point mutation is detected in the BRCA gene. Explain how a point mutation could affect the function of this gene and its implications for cancer predisposition. Answer: point mutations are a type of genetic mutation where a single nucleotide in the DNA sequence is altered. In the case of the BRCA gene, which encodes a protein essential for DNA repair, a point mutation can significantly impact the protein's function. BRCA plays a vital role in maintaining genomic stability by repairing DNA double-strand breaks through homologous recombination. A point mutation in this gene can cause a change in the amino acid sequence of the BRCA protein, potentially leading to a dysfunctional or truncated protein that cannot perform its repair function effectively. When the BRCA protein is non-functional, DNA damage accumulates, leading to genomic instability and an increased risk of mutations in other genes that may contribute to cancer development. In particular, mutations in BRCA are strongly associated with a predisposition to breast and ovarian cancers. Clinically, individuals with a BRCA mutation may undergo increased surveillance for breast and ovarian cancers and preventive measures such as prophylactic surgeries, e.g., mastectomy or oophorectomy, or chemoprevention, e.g., using selective estrogen receptor modulators, may be considered. How does a reciprocal translocation differ from a Robertsonian translocation, and what are the clinical consequences associated with each? Answer: a reciprocal translocation and a Robertsonian translocation both involve chromosomal rearrangements, but they differ in their mechanisms and clinical outcomes. Reciprocal translocation. This type of translocation occurs when two chromosomes break and exchange segments, with no genetic material being lost or gained. This means that each chromosome involved in the translocation retains its original amount of genetic material, but the genes may be rearranged. While most reciprocal translocations do not lead to a clinical phenotype in the carrier, they can cause reproductive issues, including infertility and an increased risk of miscarriage due to the unbalanced segregation of chromosomes during gamete formation. In some cases, if the translocation disrupts critical genes, it can result in genetic disorders in offspring. For example, some reciprocal translocations can lead to conditions such as chronic myelogenous leukemia, where the translocation between chromosomes 9 and 22 forms the Philadelphia chromosome. Robertsonian translocation. This is a type of chromosomal rearrangement in which two acrocentric chromosomes fuse at their centromeres, resulting in a single chromosome with two long arms and the loss of the short arms. This translocation typically involves chromosomes 13, 14, 15, 21, or 22, and can lead to Down syndrome if there is a translocation involving chromosome 21. While carriers of a Robertsonian translocation typically do not exhibit any phenotypic abnormalities, they have an increased risk of having children with unbalanced chromosome numbers, such as trisomy 21, Down syndrome. Clinical consequences include developmental delay, intellectual disabilities, and various health issues such as heart defects and gastrointestinal problems. What is the difference between a frameshift mutation and a missense mutation, and how do these mutations impact protein function? Answer: frameshift mutation. A frameshift mutation occurs when the insertion or deletion of nucleotides in a DNA sequence shifts the reading frame of the gene. This occurs if the number of nucleotides inserted or deleted is not a multiple of 3, leading to a change in the sequence of amino acids downstream of the mutation. This often results in a completely altered protein, and in many cases, the mutation introduces a premature stop codon, leading to a truncated and non-functional protein. Frameshift mutations can be highly detrimental because they drastically alter the protein's structure and function. An example of a frameshift mutation is seen in certain types of cystic fibrosis, where a small deletion in the CFTR gene results in a non-functional protein that disrupts chloride ion transport across cell membranes. Missense mutation. A missense mutation involves the substitution of a single nucleotide, leading to the replacement of one amino acid with another in the protein sequence. Depending on the location of the mutation and the properties of the substituted amino acid, the protein may still be functional, partially functional, or completely non-functional. In some cases, the mutation might alter the protein's active site or affect its stability, leading to a disease phenotype. For instance, a missense mutation in the hemoglobin gene causes sickle cell anemia, where the substitution of glutamic acid with valine alters the hemoglobin protein, causing red blood cells to sickle and impair oxygen transport. Dr. Patel is analyzing a patient's genomic DNA and detects a mutation in the telomeres. The patient has a history of early aging and exhibits symptoms consistent with a progeroid syndrome. What role do telomeres play in aging, and how could a mutation in the telomerase enzyme contribute to this patient's condition? Answer: telomeres are repetitive DNA sequences located at the ends of chromosomes, serving to protect the chromosome from degradation and preventing loss of genetic information during DNA replication. With each cell division, telomeres shorten, and once they reach a critical length, the cell enters senescence, where it can no longer divide or undergoes apoptosis. Telomere shortening is a key factor in aging and age-related diseases. Telomerase is an enzyme that adds telomeric repeats to the ends of chromosomes, counteracting the normal shortening process. It is active in germ cells, stem cells, and certain cancer cells, but it is usually inactive in somatic cells. In patients with progeroid syndromes, mutations in genes responsible for telomere maintenance, e.g. telomerase or sheltering proteins, can result in accelerated telomere shortening. This leads to premature cellular aging, tissue dysfunction, and the clinical features associated with these syndromes, such as hair loss, wrinkling, and osteoporosis. The specific mutation in telomerase would reduce its ability to maintain telomere length, leading to the rapid onset of senescence and premature aging symptoms. A common example of this is dyskeratosis congenita, a disorder characterized by premature aging, bone marrow failure, and increased cancer susceptibility. Describe the role of helicase and DNA polymerase during DNA replication, and explain how their actions are coordinated to ensure high fidelity in DNA synthesis. Answer: during DNA replication, helicase and DNA polymerase play essential roles in unwinding the DNA helix and synthesizing new strands of DNA. Helicase. The role of helicase is to unwind the double-stranded DNA ahead of the replication fork. It breaks the hydrogen bonds between the base pairs, separating the two DNA strands and creating single-stranded templates for replication. This is necessary to allow access for other enzymes such as DNA polymerase to perform their functions. Helicase moves in a 5-to-3 direction along the template strand, utilizing ATP to break the hydrogen bonds. DNA polymerase synthesizes the new DNA strand in the 5-to-3 direction by adding nucleotides complementary to the template strand. However, DNA polymerase cannot initiate strand synthesis on its own. It requires a primer, usually synthesized by primase, to provide a free 3-hydroxyl group. DNA polymerase then elongates the primer by adding nucleotides one by one. On the leading strand, DNA polymerase synthesizes continuously in the direction of the replication fork, whereas on the lagging strand, it synthesizes in short fragments known as Okazaki fragments, which are later joined together by DNA ligase. The coordination between helicase and DNA polymerase is essential for high fidelity during replication. Helicase must continuously unwind the DNA as polymerase adds nucleotides. Additionally, proofreading mechanisms within DNA polymerase detect and correct any errors in nucleotide incorporation, ensuring accurate DNA replication. This reduces the likelihood of mutations and maintains genetic stability. Explain the processes of transcription and translation in eukaryotic cells, including the roles of RNA ribosomes and the significance of post-translational modifications. Answer: transcription and translation are the two main processes involved in protein synthesis in eukaryotic cells, involving the conversion of genetic information from DNA to functional proteins. Transcription Transcription is the process by which the information in a gene's DNA sequence is copied into a messenger RNA molecule. The enzyme RNA polymerase binds to the promoter region of the gene and synthesizes an RNA strand that is complementary to the DNA template. In eukaryotic cells, the RNA undergoes several modifications before it is transported out of the nucleus, capping at the 5N, splicing, removal of introns and joining of exons, and polyadenylation at the 3N. The resulting mRNA is a mature transcript ready for translation. Translation is the process where the mRNA is decoded to synthesize a protein. The mRNA binds to the ribosome, which reads the mRNA codons. Transfer RNA molecules, each carrying a specific amino acid, match their anticodon to the mRNA codon in the ribosome. The ribosome catalyzes the formation of peptide bonds between adjacent amino acids, elongating the polypeptide chain until a stop codon is reached. After translation, proteins often undergo post-translational modifications such as phosphorylation, glycosylation, or acetylation, which are critical for protein function, stability, and localization. These modifications can activate or deactivate enzymes, facilitate protein-protein interactions, and target proteins for degradation or transport to specific cellular compartments. How do CRISPR-Cas systems work for gene editing, and what are the potential applications and ethical concerns associated with this technology? Answer: CRISPR-Cas is a revolutionary gene editing technology that allows scientists to precisely alter the DNA in living organisms. CRISPR, cluster regularly interspaced short palindromic repeats, is a bacterial immune system that uses RNA molecules to guide the Cas protein to specific sequences of DNA. Cas then cuts the DNA at a designated location, enabling either the addition, removal, or modification of specific genes. The CRISPR-Cas system works by designing a guide RNA that is complementary to a target DNA sequence. This guide RNA directs the Cas protein to the exact location in the genome where the cut should be made. Once the DNA is cut, the cell's repair mechanisms are triggered, which can be harnessed to introduce desired genetic changes. This can involve correcting mutations in genes, inserting new sequences, or disabling unwanted genes. Applications of CRISPR-Cas are vast, ranging from medical therapies such as gene therapy for genetic disorders, e.g., sickle cell anemia, to agricultural improvements such as developing crops resistant to diseases or environmental stress. CRISPR is also being used in biomedical research to create animal models for studying diseases. What is the principle of FISH fluorescence in situ hybridization, and how can it be used to detect chromosomal abnormalities? Answer: fluorescence in situ hybridization, FISH, is a cytogenetic technique that uses fluorescently labeled probes to detect and localize specific DNA sequences on chromosomes. The principle of FISH involves hybridizing a DNA probe, which is complementary to the target sequence of interest, to chromosomes in a metaphase or interphase cell. Once bound, the probe emits fluorescence when exposed to ultraviolet or visible light, making it visible under a fluorescence microscope. FISH is commonly used to detect chromosomal abnormalities such as translocations, deletions, duplications, and aneuploidy. For example, in diagnosing Down syndrome, FISH can be used to identify an extra copy of chromosome 21. In cases of chronic myelogenous leukemia, CML, FISH can detect the Philadelphia chromosome, a reciprocal translocation between chromosomes 9 and 22. One of the major advantages of FISH is its ability to analyze specific genes or chromosomal regions without the need for the entire chromosome to be visualized. Additionally, FISH can be performed on non-dividing cells, making it suitable for analysis of tissue biopsies or prenatal samples. How do G-banding and Q-banding differ in cytogenetic analysis, and what are the advantages of each technique? Answer: G-banding and Q-banding are both chromosome staining techniques used to visualize chromosomes and their banding patterns under a microscope, but they differ in the methods and types of patterns they produce. G-banding. This technique uses genes sustained to produce a pattern of light and dark bands along the chromosomes. The dark bands typically represent regions of the chromosome that are rich in adenine thymine at base pairs, while the light bands are rich in guanine cytosine, GC, base pairs. G-banding provides a consistent and reproducible banding pattern across chromosomes and is widely used for karyotyping. It helps in identifying large-scale chromosomal abnormalities such as deletions, duplications, and translocations. Q-banding. In this technique, a fluorescent dye such as quinacrine is used to bind to DNA, and the chromosomes are viewed under ultraviolet light. The banding pattern produced by Q banding is different from that of G banding, with brighter bands indicating regions with a high AT content. Q banding is particularly useful for detecting small chromosomal abnormalities that may be difficult to identify with G banding. It also provides better resolution in some cases, making it more suitable for studying structural changes like inversions and small deletions. A child is suspected of having Fragile X syndrome due to developmental delays and facial features associated with the disorder. The pediatrician orders a genetic test. Which test is most appropriate for confirming the diagnosis, and what is the underlying molecular mechanism detected by this test? Answer: the most appropriate test for diagnosing Fragile X syndrome is FSR gene analysis using Southern blot or PCR to identify the number of CG repeats. Fragile X syndrome is caused by an expansion of the CG trinucleotide repeat in the promoter region of the FSR gene located on the X chromosome. Normal range: fewer than 45 CG repeats. Premutation range: 55200 repeats. Associated with Fragile X-associated tremor-ataxia syndrome (FSTAS) or Fragile X-associated primary ovarian insufficiency (FXPOI). Full mutation: more than 200 repeats, leading to hypermethylation of the FSR promoter and silencing of the gene, resulting in the absence of FMRP. Fragile X mental retardation protein, which is crucial for synaptic plasticity. Relevant guidelines: the American College of Medical Genetics and Genomics recommends FSR gene testing as the first-line diagnostic approach in males with intellectual disabilities, developmental delay, or autism spectrum disorder. Explain why mitochondrial diseases such as ME, LAS, and Lee syndrome often exhibit varying phenotypes, even among individuals with the same mutation. Answer: mitochondrial diseases exhibit phenotypic variability due to several factors. Heteroplasmy: mitochondria within a cell may have a mix of normal and mutated DNA. The ratio of mutated to normal and DNA determines disease severity. Threshold effect: different tissues require varying levels of mitochondrial function. Energy-demanding tissues like the brain and muscles are more affected. Nucleomitochondrial interactions: proteins encoded by nuclear DNA influence mitochondrial function and can modify the phenotype. Environmental and lifestyle factors: factors such as diet, stress, and coexisting conditions can impact the severity of mitochondrial diseases. Relevant guidelines: ACMG recommendations include heteroplasmy quantification during genetic testing and considering family history in genetic counseling. Dr. Emily is managing a patient with a confirmed diagnosis of a neurodegenerative disorder caused by a loss-of-function mutation in a single gene. The disorder is progressive, with significant motor impairment and cognitive decline. Gene therapy is being considered. What factors should Dr. Emily assess before proceeding, and what are the potential risks and ethical considerations of using gene therapy in this case? Answer: factors to assess. Gene delivery system: evaluate whether an adeno-associated virus or vector is appropriate for the specific disorder. Target cell type: ensure the therapy targets neurons or glial cells which are most affected in neurodegenerative conditions. Timing: earlier intervention may be more effective to prevent irreversible damage. Duration of expression: determine if a single dose will provide sustained benefit or if repeat dosing is required. Risks and ethical considerations. Off-target effects: potential for unintended genetic modifications in non-target cells. Immune response: risk of an immune reaction to the vector. Equity and access: high costs of gene therapy raise concerns about accessibility. Informed consent: ensure patients and families fully understand the experimental nature and risks involved. Compare the purposes and methodologies of pre-implantation genetic testing for aneuploidy, PGE, and pre-implantation genetic testing for monogenic disease PGE. Answer: PGT aneuploidy. Purpose: screens embryos for chromosomal aneuploidies, e.g. trisomy 21, to improve implantation rates and reduce miscarriage risk. Methodology: uses technologies like array comparative genomic hybridization or next-generation sequencing (NGS) to assess the chromosome copy number. PGOM, monogenic diseases. Purpose: detect specific single gene mutations in embryos for conditions like cystic fibrosis or sickle cell anemia. Methodology: uses targeted analysis of the parental mutation via PCR and linkage studies to avoid transferring embryos with the disease-causing mutation. Relevant guidelines: ESHI, European Society of Human Reproduction and Embryology. Guidelines recommend using PGE for advanced maternal age or recurrent pregnancy loss and PGE when one or both parents are carriers of a monogenic disorder. Mrs. Taylor, a 35-year-old woman, is 12 weeks pregnant. Her obstetrician suggests cell-free dscDNA testing as part of her prenatal care. Mrs. Taylor asks about its accuracy and whether it replaces invasive tests. How should her physician counsel her? Answer: counseling points. Accuracy DNA testing has high sensitivity and specificity for detecting common aneuploidies, e.g. trisomy 21, 18, 13. It is a screening test, not diagnostic. Invasive testing cDNA testing reduces the need for invasive tests like amniocentesis but does not replace them. Confirmatory testing is required for positive results. Limitations: it cannot detect all genetic disorders such as single gene defects or structural anomalies. Relevant guidelines: the ACOG recommends cDNA screening for all pregnant women, emphasizing informed consent and understanding of its limitations. A 3-year-old girl presents with loss of purposeful hand movements, repetitive hand wringing, and developmental regression following a period of normal development. Genetic testing reveals a mutation in the MEP gene. What is the molecular mechanism behind Rett syndrome, and why is it specific to females? Answer: Rett syndrome is caused by mutations in the MEP gene, which encodes a protein that is involved in regulating the expression of other genes by binding to methylated DNA. In individuals with Rett syndrome, a mutation in the MEP gene leads to a loss of function of this protein, disrupting normal brain development, synaptic plasticity, and neuronal function. Why specific to females? Rett syndrome primarily affects females due to the X-linked nature of the MEP gene. Females have 2 X chromosomes, so in most cases, one healthy X chromosome compensates for the defective MEP gene in early development. However, in males, the single X chromosome carries the mutation, and they typically die in utero or shortly after birth due to the severity of the disorder. Relevant guidelines: ACMG guidelines recommend genetic testing for MEP mutations in females with unexplained developmental regression or signs of Rett syndrome. Schizophrenia is a multifactorial disorder with both genetic and environmental influences. Explain the concept of genetic predisposition in schizophrenia and how it interacts with environmental factors. Answer: schizophrenia is thought to arise from a combination of genetic predisposition and environmental factors, a model known as multifactorial inheritance. Genetic factors: there are several genes associated with schizophrenia, such as those involved in dopaminergic and glutamatergic neurotransmission, but none are sufficient to cause the disorder on their own. The heritability of schizophrenia is estimated to be around 80%, suggesting a strong genetic influence. The COMT and DISC genes are among the most studied. Environmental factors: events during prenatal development, such as maternal infection, malnutrition, or stress, can increase the risk of developing schizophrenia later in life. Other triggers include drug use, e.g. cannabis, stressful life events, and urban living conditions. Gene-environment interaction: the interaction between genetic susceptibility and environmental exposures is often referred to as the gene-environment interaction, where environmental triggers can unmask or exacerbate the effects of a genetic predisposition. Mr. Roberts, a 38-year-old male, has been diagnosed with azoospermia. His physician recommends genetic testing, including testing for Y chromosome microdeletions. Mr. Roberts is concerned about the impact of this diagnosis on his fertility options. How should the geneticist explain the results and counseling options to Mr. Roberts? Answer: explanation of Y chromosome microdeletions. Y chromosome microdeletions can lead to male infertility, especially in men with azoospermia or oligospermia, low sperm count. These deletions involve the AZF, azoospermia factor region of the Y chromosome, which is crucial for sperm production. There are 3 regions, ASFA, ASP, and deletions in each region have different implications for fertility. ASFA or ASP deletions: these are more severe and typically result in complete absence of sperm production. ASP deletions: these may lead to reduced sperm production but not complete absence. Counseling options: if AZF deletion is present, Mr. Roberts should be informed that assisted reproductive technologies such as testicular sperm extraction, teeth, and intracytoplasmic sperm injection may be options, but the likelihood of obtaining viable sperm may be low. Carrier status: if Mr. Roberts has a Y chromosome microdeletion, he will pass the genetic defect to all of his male offspring, potentially affecting their fertility as well. What are the primary ethical concerns surrounding pre-implantation genetic testing (PGT), and how should genetic counselors address these concerns with prospective parents? Answer: the primary ethical concerns surrounding PGT include embryo selection versus genetic discrimination. PGT allows for the selection of embryos based on genetic traits, which can raise concerns about the potential for eugenics. The idea of selecting embryos based on desirable traits, not just for preventing disease. Consent and autonomy. There are concerns about whether parents are making informed decisions for their children as embryos are selected before they have a chance to develop, potentially leading to ethical dilemmas about the future child's life and autonomy. Psychosocial implications. Parents may feel emotional or moral distress about the potential for discarding embryos that do not meet genetic expectations or for selecting embryos that may carry inheritable conditions. Addressing concerns. Genetic counselors should emphasize that PGT is a preventative tool designed to reduce the risk of transmitting serious genetic disorders, rather than a means of ensuring a designer babies. Informed consent is crucial. Parents should fully understand the limitations, risks, and options available in PGT. They should also be made aware of the potential implications of selecting or discarding embryos. Dr. Johnson is counseling a pregnant woman, Mrs. Clark, who has been taking an over-the-counter medication containing an undisclosed teratogen. Mrs. Clark is concerned about the potential impact on her baby. How should Dr. Johnson approach this situation considering both genetic and environmental factors? Answer: risk assessment. Timing of exposure. The timing of exposure to the teratogen during pregnancy plays a critical role. The first trimester is the most critical period for major organ development, and exposure to teratogens during this period has the highest risk of causing birth defects. Type of teratogen. The effects of the teratogen depend on its mechanism of action. Some teratogens cause structural abnormalities, while others may lead to functional defects or growth retardation. Genetic susceptibility. Some individuals may be genetically predisposed to increased sensitivity to specific teratogens due to differences in metabolic enzymes, transporters, or DNA repair mechanisms. Approach. Dr. Johnson should review the FDA pregnancy categories for the drug involved and consider alternative options for Mrs. Clark. A teratogen exposure history should be taken, and genetic counseling may be warranted if there are concerns about hereditary susceptibility. An ultrasound and screening test should be considered to monitor fetal development. A pregnant woman undergoes non-invasive prenatal testing (NIPT), which indicates a high risk for trisomy 18. However, her ultrasound findings show no anomalies. How should the next steps in this case be managed considering the potential for false positive results in NIPT? Answer: NIPT is highly sensitive for detecting chromosomal abnormalities, but is not diagnostic. In this case, given the high-risk result for trisomy 18, confirmatory testing such as amniocentesis or chorionic villus sampling (CVS) should be performed to validate the NIPT results. Rationale: false positives can occur due to confined placental mosaicism or maternal chromosomal abnormalities. Ultrasound findings can complement NIPT, but cannot independently confirm or refute a diagnosis of trisomy 18. Relevant guidelines. The American College of Obstetricians and Gynecologists (ACOG) recommends confirmatory diagnostic tests following positive NIPT results. The use of ultrasound to identify associated anomalies such as intrauterine growth restriction or cardiac defects should guide further clinical decisions but not replace diagnostic testing. Explain the role of whole genome sequencing in diagnosing rare genetic diseases and discuss its limitations in clinical practice. Answer: WGS provides a comprehensive analysis of the entire genome, identifying variants in coding and non-coding regions. It is particularly effective in diagnosing rare diseases that are challenging to detect with targeted panels or exome sequencing. Advantages: broad scope, can identify single nucleotide variants, structural variants, and copy number variations. Diagnostic yield. Studies show a 25.50% diagnostic rate in undiagnosed rare disease cases. Limitations: incidental findings may uncover unrelated genetic risks, requiring ethical counseling. Interpretation challenges: variants of uncertain significance, thus can complicate diagnosis. Cost and accessibility: high costs and limited availability can restrict use in resource-poor settings. Relevant guidelines. The American College of Medical Genetics and Genomics recommends WGS as a second-tier test for undiagnosed disorders after targeted testing fails. Mrs. Garcia is 30 weeks pregnant, and her ultrasound reveals features consistent with skeletal dysplasia. A follow-up genetic panel identifies a mutation in the FGFR gene associated with achondroplasia. She is concerned about her delivery options and the potential health complications for her baby. What are the appropriate next steps in managing this pregnancy, and what should be communicated to Mrs. Garcia about delivery and immediate neonatal care? Answer: management plan, delivery planning, coordination with a multidisciplinary team including obstetrics, neonatology, and genetics is essential. Vaginal delivery may be feasible but could be complicated by macrocephaly. Cesarean delivery should be considered if cephalopelvic disproportion is identified. Immediate neonatal care. Close monitoring is required for potential respiratory complications due to a small chest cavity. A detailed birth plan should include access to a neonatal intensive care unit (NICU). Counseling points: educate Mrs. Garcia on achondroplasia's typical prognosis, including expected growth and developmental milestones. Discuss the risk of foramen and magnum compression and other long-term complications. Relevant guidelines. Guidelines from the American Academy of Pediatrics recommend specialized monitoring for newborns with skeletal dysplasia to address airway and neurological concerns immediately postpartum. What are the key considerations when integrating ultrasound findings with non-invasive prenatal testing (NIPT) for a comprehensive risk assessment? Answer: key considerations. Correlation of findings. Concordant results e.g. both NIPT and ultrasound indicating anomalies increase the likelihood of a true positive. Discrepant findings. Require detailed counseling and further diagnostic testing. Maternal factors. Conditions like obesity or twin pregnancies can reduce NIPT accuracy and ultrasound resolution. Timing of ultrasound. Sequential scans, first and second trimester, provide better detection rates for structural anomalies. Relevant guidelines. The International Society for Prenatal Diagnosis emphasizes the combined use of NIPT and ultrasound to enhance screening sensitivity and specificity. Discuss the challenges in developing and approving orphan drugs for rare genetic diseases, focusing on the incentives provided by regulatory agencies. Answer: challenges: small patient populations, limited data for clinical trials, high R&D costs, difficulty in recouping investments due to low demand. Regulatory incentives. Orphan Drug Act 1983 provides tax credits, grant funding, and 7 years of market exclusivity in the U.S. European Medicines Agency (EMA) offers fee reductions and market exclusivity for 10 years. Fast-track approvals. Programs like FDOS Breakthrough Therapy Designation expedite access for critical treatments. Why is pre-implantation genetic testing (PGT) crucial during in vitro fertilization and what ethical considerations arise from its use? Answer: pre-implantation genetic testing (PGT) is a crucial tool in vitro fertilization as it allows for the screening of embryos for genetic conditions before they are implanted in the uterus. This process helps to identify embryos that may carry inheritable genetic disorders, such as cystic fibrosis, Huntington's disease, or Down syndrome. It significantly increases the chances of a successful pregnancy without passing on genetic diseases to the offspring. PGT can also identify chromosomal abnormalities like aneuploidies, extra or missing chromosomes, which are linked to conditions such as trisomies 13, 18, and 21. This helps to prevent miscarriages due to such chromosomal defects. Ethically, PGT raises several considerations. There is concern over design at babies of where genetic selection could extend beyond preventing disease to selecting non-medical traits such as intelligence or physical characteristics, potentially leading to a future of eugenics. Additionally, the availability of PGT raises questions of equity as it may only be accessible to wealthy individuals, exacerbating healthcare disparities. Another ethical dilemma is ensuring proper informed consent from patients, who must fully understand the implications of using PGT, including the potential for incidental findings that might not have been anticipated. Compare the roles of metabolomics and genomics in diagnosing rare diseases. Answer: both metabolomics and genomics play critical roles in diagnosing rare diseases, but they focus on different aspects of disease pathophysiology. Genomics involves analyzing an individual's DNA to detect genetic mutations that may lead to rare diseases. By identifying specific mutations, such as those in cystic fibrosis transmembrane conductance regulator CATR for cystic fibrosis or FBN for Marfan syndrome, genomics can provide a precise diagnosis of hereditary conditions. Genomic testing, including whole exome sequencing, or whole genome sequencing, is particularly helpful when a patient presents with undiagnosed conditions, especially when traditional testing panels fail. On the other hand, metabolomics involves studying the metabolic profile of an individual, focusing on the small molecules present in blood, urine, or other tissues. It can reveal metabolic disturbances caused by genetic mutations. For instance, in inborn errors of metabolism, such as phenylketonuria PQ or maple syrup urine disease, MSUD metabolic profiling can help detect abnormal levels of metabolites. A metabolomics is also valuable when genetic testing is inconclusive, as it can provide complementary data to support or challenge a genetic diagnosis. Dr. Roberts is managing a child with suspected Bardet-Biedl syndrome (BBS) presenting with obesity, retinal dystrophy, and polydactyly. The family inquires about genetic testing and prognosis. How should Dr. Roberts proceed in confirming the diagnosis and counseling the family? Answer: confirming diagnosis. To confirm the diagnosis of Bardet-Biedl syndrome (BBS), Dr. Roberts should order genetic testing for mutations in the BBS genes. The most common method is whole exome sequencing, or targeted genetic panels that screen for the key mutations in the BBS-associated genes. BBS is an autosomal recessive disorder, meaning the child inherits 2 copies of a defective gene, one from each parent. In addition to genetic testing, Dr. Roberts should assess the clinical features systematically. BBS is characterized by a combination of obesity, retinal dystrophy, leading to progressive vision loss, polydactyly, extra fingers or toes, and often kidney anomalies and developmental delays. Counseling the family. Once the diagnosis is confirmed, Dr. Roberts should provide genetic counseling to the family, explaining the inheritance pattern, autosomal recessive, and the implications for future children. The family should be informed about the prognosis, which varies based on the severity of the symptoms. Support for the family is crucial, as BBS can significantly affect a child's quality of life. Dr. Roberts should offer resources for family support groups and patient advocacy organizations. What are the implications of detecting microdeletions through prenatal testing, and how should such findings be addressed? Answer: microdeletions refer to small chromosomal deletions that are often too small to be detected by conventional karyotyping, but they can be identified through more advanced genetic testing techniques like array comparative genomic hybridization or fluorescence in situ hybridization (FISH). Prenatal detection of microdeletions raises important clinical and ethical implications. Clinical implications. Microdeletions can be associated with a range of syndromic conditions, such as DiGeorge syndrome, 20.2 deletion, Prader-Willi syndrome, and Angelman syndrome. These conditions often result in physical and intellectual disabilities, as well as other health complications such as immune system deficiencies, heart defects, and developmental delays. In some cases, the detected microdeletion may not result in a recognizable syndrome, or its effects may be variable, leading to uncertain prognostic outcomes. The uncertainty regarding the clinical impact of the deletion is a significant challenge in counseling families. Addressing findings. Upon detection of a microdeletion, it is essential to confirm the findings with diagnostic testing such as amniocentesis or CVS if not already performed. This will provide a more definitive diagnosis. Genetic counseling is crucial to help the parents understand the implications of the microdeletion, including the potential for an impaired quality of life for the child, possible long-term care needs, and options for future pregnancies. The counselor should also explain the inheritance pattern and discuss options for preconception counseling, such as pre-implantation genetic testing (PGT) for subsequent IVF pregnancies. If the microdeletion is linked to a syndrome with known management protocols, the family should be referred to specialists in the relevant areas such as cardiology, nephrology, and endocrinology to develop a care plan. For conditions where the outcome is uncertain, a multidisciplinary approach to care may be necessary, involving geneticists, pediatricians, and developmental specialists. Outline a multidisciplinary approach to managing patients with Ehlers-Danlos syndrome (EDS). Answer: Ehlers-Danlos syndrome (EDS) is a group of connective tissue disorders characterized by hypermobility of the joints, skin hyperelasticity, and fragile skin that bruises easily due to the variable nature of EDS. A multidisciplinary approach is essential for optimal management, as the condition can affect multiple organ systems. Core team members, geneticists, to provide a definitive diagnosis, assess the type of EDS. There are several subtypes, and offer genetic counseling regarding inheritance patterns, often autosomal dominant. Cardiologist. Some forms of EDS, particularly vascular EDS, can affect the blood vessels, leading to a risk of aneurysms or vascular rupture. Regular cardiac evaluations and monitoring of the vascular system are necessary. Orthophysical therapist. Many patients with EDS have joint hypermobility, which increases the risk of dislocations and other musculoskeletal issues. Physical therapy can help strengthen muscles to provide stability to hypermobile joints and prevent injury. Dermatologist. To address the skin issues, including its fragility and scarring tendency, dermatologists can help manage wound healing and avoid skin trauma. Focus areas. Pain management. Many individuals with EDS experience chronic pain due to joint instability and soft tissue damage. A multidisciplinary team, including pain specialists, can provide medication, physical therapy, and possibly psychological support to manage chronic pain. Surgical caution. Surgical procedures should be approached with caution due to the tendency of patients with EDS to have delayed wound healing, scarring, and fragile tissues. Careful consultation with a surgeon familiar with EDS is crucial before any invasive procedures. Patient education. Genetic counseling and patient education on avoiding trauma, understanding their condition, and coping strategies for managing chronic conditions are essential for empowering the patient and family. Through this multidisciplinary approach, the healthcare team can provide a personalized and comprehensive care plan addressing both the immediate concerns and long-term management of the condition.